images everywhere, also in NLP, with a lot of new thing that appears, providing extremely good results in many different data sets that have provided a lot of interest from the industry for the many applications in biology, in medicine, like we will see during this presentation, but of course in many other domains. There is, however, few limits to deep learning, even though some are starting to be addressed and with some solution, especially the first one, that it's necessary to have a lot of data in order to be able to make this approach work. When we are talking about images, it requires annotation, and in the medical field, it means that you need an expert, a doctor that is usually very busy to annotate the data set in order to be able for the models to train on. Another problem, and it's also the case in particular in medicine, we have deep learning is usually called black box approach. It means that you cannot understand how the decision is taken inside the model. We'll see some ways of trying to understand how the model is working, but still a big limitation in my opinion. Also, and I will not address that in this presentation, but we have worked in the team going in this direction. It's consuming a lot of energy to train that. We need big machine, big graphic cards that require electricity. And this, the question is that approach sustainable in the future when we know that the resources are limited on Earth? So we need to work on this kind of problems. Very quickly, what is deep learning? Deep learning is a huge network, and I will not enter into the detail on how it's working. It's a huge network of components that are called neurons that are looking like that are very simple computational units where you do the sum of inputs here, weighted inputs, the synaptic weights, that goes into an activation function, and you make a big network out of it. And this allows to take complex decisions. Deep learning is when you have different layers that can be specialized in certain treatment, like convolutional layers that are able to compute local pixel analysis to extract some feature. And then you have a classifier at the end or whatever kind of head. The idea here is that you will really need millions of billions of parameters in order to make this work, to train them on the data set. And this is what makes the approach black box. It's because you have this high number of neurons, high number of synaptic weight that makes the decision hard to explain. There is a full field of research. I will provide some example during the presentation of that, named explainable AI, that tries to understand how neural networks are working, but not yet sufficiently interpretable in my opinion. This need of interpretability is also necessary because of the new rules, especially in Europe. We have an AI Act that has been voted a few years ago, that in critical domains, including transportation or medicine, where the life of human can be impacted by the decisions that are taken by AI system, including deep learning, where you need, so if I take the EU AI Act, we need to have algorithms that are transparent, comprehensible by users, meaning that we need to understand how the decision is taken to be sure that the decision is taken for right reason before applicable. And even if explainable AI came way before this AI Act, it reinforces this necessity to understand how the decision is taken. What is interesting is that in the FDA, which is the American version of the health system, we are talking about interpretability and not about explainability. And that needs to be clarified because there is actually in the literature a lot of people that are exchanging these two words, explainability and interpretability, whereas it's not the same meaning when we are talking about these two terms. So if I give the definition of explainability, it's the analysis of the behavior of the model. Basically, it's most of the time done on the a posteriori. It means that we are trying to know what are the inputs that have been used in order to take the decision. One good example of that when we are doing image analysis is when we are trying to segment this image with a dog and a cat. We can see what are the pixels that has been used in order to take the decision that the cat is here, so it's the red pixel here. Or with dog, it's using the face of the dog in order to take the decision to say that the dog is here. So this is a very classical in the explainable AI field, a very classical method, sorry, in the explainable AI field that is called GradCam. The idea is that here we can see the inputs that are used. Here we can say that the model is taking the decision using the right inputs, and it's the same here for the dog, but we don't know how the model decided that this is a cat here and this is the dog. We don't know if he took the decision because a cat is always lying on the floor because it's a lazy animal, for example, and the dog is always standing up, very proud of him or her, actually. So we don't know how the decision has been taken. And that's the aim of interpretability. The interpretability is when you want to know the full mechanism of the decision process, meaning like you want to understand how the inputs are processed step by step, one by one, function by function, until taking the final decision. And you understand the full flow of decision that is used by the model in order to take the final decision. And we have systems in machine learning that are already fully interpretable. Linear regression, you can look at the coefficient, for example, in your linear regression to see how the decision is taken. Decision trees, look at the tree itself, see the threshold that are used to go from either on left or right branch, for example, or a rule-based system such as a classifier system that we were talking about, where you can look at the rules and look how it's written. So we have already things that are working. Unfortunately, these algorithms are usually less performant than deep learning. And what we would like is to have some algorithms that are interpretable, meaning I can understand everything from the input to the output, all the decisions, how they are taken, and also efficient to solve the task. So in the team, we are working on the alternative approach to deep learning that is called genetic programming. And the idea of genetic programming is to actually reuse things that have been developed in order to do image processing, if I stay in this example of image processing. So basically, when you are looking at the deep learning and you take a convolutional layer, each convolution filter is trying to compute, extract some feature from the image and recreate potentially a function that exists in image processing. Like, for example, if I take this image here, you have coins. I don't know if you can see them correctly here. And you have a function that is called a Sobel filter that is taking the edge of each object. And it's a convolution that is applied in this case. So the Sobel filter could be reinvented by deep learning, whereas it has been already mathematically done before, and it exists in a function library. So the idea of genetic programming is actually to try to reuse these decades of engineering in image processing and try to automatically assemble and optimize all these functions, how we use them all together, how we build automatically a pipeline based on this function in order to process the image, to process the data, in order to take the right decision. And because it's an assemble of existing functions that are well described in the function library, it means that the program that are generated should be, I'm saying should be for now, interpretable in the sense that I can read the code and understand how the decision process is taken. So let me go quickly on the theory that is behind. So genetic programming are based on genetic algorithms, and I'm going to introduce them quickly. So these algorithms are stochastic optimization algorithms that are inspired by the Darwinian evolution. So this idea of trying to encode solution into a genome, make a population of them, it's the P0 that you have here. You evaluate how fitted each individual of the population is for a given task. Whatever the task is, you have a function that is scoring each individual. You do a selection, meaning that you select the fittest individual, trying to get also some diversity in your population. And these are allowed to be modified by mutation, so randomly modifying some part of the genome, or by crossover, meaning that you can cross subparts of the genome. And you do that for a certain amount of time, repeating this process. And by selective pressure, you obtain a good solution to your process. So to give you quickly an example of that, it's a very famous, it's one of the first industrial success of GA 20 years ago now. It's actually the anniversary of that. Actually, it's this antenna that you can see on the top right of the image of the screen is an antenna that is in the ISS, I think it is in English as well, whatever. And it has been optimized by the genetic algorithm. So what you do, you encode inside the genome the size of each branch, the angle that you have for each rotation here. So it's way more number that are here. And you evaluate them using a simulator, what's the quality of the reception that you get with this kind of antenna. It allows you to select some of the best ones. So here's the best grades that I have put. And then that's your subpopulation. You can select a subset of them and apply a crossover. So crossover, I took, for example, these two first numbers for this antenna. That could be the bottom of the antenna and the top of the other antenna, of the green one. And you combine them and you obtain a new antenna that you hope is more fitted. And you can do mutation. So you select one individual, you apply the random mutation. So for example, that could be the elongation of the third line, let's say, and you elongate it randomly of a given value. And that you obtain a new population that is getting the traits of your previous population with some, hopefully, improvement that will be selected and crossed and mutated again in the future. In genetic programming, it's exactly this process that is applied, but to programs. So we can encode programs, for example, in a tree here. So your genome is actually this tree that is representing this mathematical function here. You have the plus that is on top, is this plus here. The minus is here. 2.2, it's a constant that can be optimized. Everything, all this program of this mathematical function is encoded inside a genome, and you apply evolution. So you evaluate that against this task. You select the best. You modify them. To modify, you can change this operator minus here by a star, and it's going to change a multiplication here in your equation. You can cross two graphs, take this subgraph here, cross it with another one, and take the other subgraph and put it here, and I will change all this part of the equation. So evolution can be applied exactly as I showed on this basic scalar optimization to programs, to mathematical function at the end. So I will show how it works on the image just after. But before, I would like to get back to interpretability and how we can make this approach interpretable. So in order to make a decision process interpretable, we need three components and I will focus on two of them today. The first component is simulability. So simulability is the capacity of human to reproduce the decision process inside the brain. Like if I take an example of that, how I can imagine and I can imagine how each step of the decision process has been used and applied and how it impact the decision at the end. Decomposability, it's in order to understand how complex decisions are taken, the best way is to be able to decompose the decision process into sub-components that are simpler to understand and potentially, again, decomposed until I have atomic operators that I can interpret. And then I can go back to the decision and understand how the complex one is taken. And transparency, that is common to any machine learning approach, in my opinion, is to guarantee that we have a converging process during learning, meaning that I know that I will obtain a good model and I know what kind of data I've been training on, the quality of the data, and so on and so forth. So things that is very common in machine learning approach. So let's start with simulability. So again, the capacity of the human of a human brain to reproduce and imagine the decision process in the head. So basically, a neural network is interpretable, is simulable. You can decompose a neural network saying, I have this weight, I have my transfer function, my weighted sum, I have my activation function here, and I can imagine for each neuron what was the decision. I can compute in my head the activation step, the activation value, pass it to the next neuron. I could do that. The problem is that you have billions of neurons, billions of weights, and it's impossible for a human, or very complex, let's say, very complex, maybe not impossible, but very complex, and it would take forever to be able to process the full neural network, to do all the computation to understand how it's done. And this is why we are usually using these post-hoc operators in order to understand, to see what are the inputs that are used and not the full decision process. The problem with that is that it's showing where the decision is taken. So the cat is here, the dog is here, but we don't know why. We only imagine. I only imagine that these inputs are used here and it's a cat because a cat is a lazy animal and it's always lying on the floor, and then it's a cat. But that's what I imagine. Maybe the neural network is saying this is a cat because it's long, and that's it. I don't know. I have no clue how the neural network is taking the decision here. And it can induce error in the explanation because it's what I imagine from the neural network based on this pixel only. In genetic programming, actually, because we have function, we can see how the decision is taken. I will not explain that in detail inside each graph, but if I do the parallel between an image that is processed by a neural network and with explanation, so this is what we call histopathological images, so it's a sample, a tissue sample that you have to classify as cancer or non-cancer, basically. You can see the cells, it's the black dots that you have everywhere. If I pass that to a ResNet 18, whatever neural network, it's a deep learning neural network that provides good results, let's say, in this data set, I can see highlighted what are the cells that are used in order to take the decision. But that's it. I know that these cells here is used to process, but I don't know why this one is not used, which is pretty much the same. I don't know how the cells are taken into consideration here. Whereas if I use a genetic programming approach, I'm training on the same data set. I will show that later. We obtain comparable results in terms of efficiency, but at the end, I have this kind of graph here. And I can look at what are the inputs that are used, so the different channels that I have here, and how they are processed. So for example, here to get you a quick idea of how it's using, It's calculating, so it's trying to make mask of each cell. It's what you see here. It's the cells that you have. And then at the end, the reduced N color here is used to count how many cells you have in the image. And basically, if you talk with a doctor, and we did this work, it's using the density of cells. The denser the tissue, the riskier it is in terms of cancer. So the more cells you have here, the decision is taken like that. The more cells you have, the more risk you have to be cancerous. Decomposability, yeah. So at least, sorry, I forgot the conclusion here. Simulability here, I was able to explain because I know what the Laplacian is doing here. I know what the mask is doing. I have the full description of the code. I can look at the code. I can understand how it's processed, and I can imagine what it does on the image. If I take another image of my data set here, I can imagine what would be the output of this function here and how it's applied, how it's going to work. So I can understand, I can simulate how the decision process is taken in my graph. Another important component in interpretability is decomposability, so the capacity to decompose the decision process in subsets that are in sub-components that are easier to understand. And when you think about it, it's exactly what we are doing in every decision system, or every single system that you have in the society. So if I take a complex piece of software, so if, for example, here it's an image of the electronic health system, a record system from an hospital in the US, I can see many different components. So with my database that is storing the data, I have the clinical record that is here, a view that is pharmacy, I have another view for the radiology. And even if I have a huge system, I can explore only this part here, the radiology system, for example, understand how it's working, and I can understand how it's working, how it's connected to the single clinical data repository, which is connected to the database. So I can understand how everything is intertwined in the system, and understand the system in its globality and also in a very local part. So that's this way of organizing software is necessary because it's the only way to do good maintenance in this system, because you can, if you have a bug here, it should be only here and not diffusing everywhere. So you have a system that is more robust. You can do certification also, certificability, because you can certify each component separately and then assemble and certify the assembly. And like I said in example, you have an interpretable system, because even if you have a huge code, you can decompose the code as the per instance. And when you are computer scientists, it's actually what you are doing in your code. You are building a function that is based on simple operators. You understand the function as a whole. You know what it does. You can write the documentation about the input, the output, how it's working, what it's processing. You don't have to understand this code. And you can build on top of that another function that is calling this function without understanding the code anymore. But you can understand how this function is used, how it's intertwined, and so on and so forth. So this level of abstraction is necessary to build more complex decisions. Basically, we have different levels of decomposition. So the function level that I talk about, you have small components that are used in function that you regroup. You know how it's working. You know what it does. You do by type also, being able to follow the data types, the fact that you have patient data, are they clinical, are they images? You are not going to process this different type of data of the same way. So it's important to understand how the types are processed and being able to follow these data types. And by decision, so how, again, all these components are assembling together in order to understand the big picture of the decision process. And it's the same, actually. I can draw the comparison between genetic programming and and neural networks. Neural networks are decomposable. Again, I have one neuron. One neuron is super simple to understand how it's working. Again, transfer function with my weighted sum of the inputs and the activation function. If I do that very basically, that's easy. That's a simple decomposition. But the problem is that it's, again, very intertwined. Like every single neuron is connected to thousands of neurons, and you have a lot of connection. That makes the composition useless because you cannot follow the flow of decision at the end. You could have the same issue with genetic programming. You could obtain equation like that. That doesn't mean anything, so it's been generated by a LLM, so it really doesn't do anything. But you cannot understand how it's working because everything is intertwined. So the idea is how I make interesting decomposition in order to be able to understand the decision process. And it's what we did actually for this first application about biomedical application. The idea was to be at the function library in genetic programming. So because genetic programming is assembling function, you need to develop a function library that GP will assemble after. And you need to have functions that you decide that are taken. And the big thing that we proposed is instead of only using min, max, add, subtract, and very simplistic operators that are commonly used in genetic programming, to take one level of abstraction and propose more complex functions that exist in OpenCV, for example, for image processing. Functions that I know that are doing morphological operation, other that are working on the mask at the mask level. So functions that are composed at the end of a lot of add, multiplication, division, and so on and so forth, but are already abstracted with this idea of being able to read the pipeline at the end by knowing what each function is doing. What we obtained at the end, and it was one of the first good results that we obtained, was on image segmentation. So we had a benchmark composed of 89 images of this type here that you have. So you have this is a cell nucleus, and you need to segment each nucleus, meaning that you need to find a mask that corresponds to each cell in this image. And that's a task where neural networks are very good, in particular at the time. So it's a work that is 3 years old now. There was a new neural network named CellPose that has built a data set for that was performing very well in comparison to the state of the art. It was trained, so CellPose is trained on the tens of thousands of images, and then fine-tuned on this specific data set of 100 images, 89 images for training, and 11 kept for testing. So we compared GP, so it's a specific genetic programming approach called Cartesian genetic programming. I will not enter into these details. With on this data set, and what we observe is that, so this is the performance of CellPose and two other neural nets, so Masker CNN and StarDist, that are, let's say, classical segmentation neural nets. And what we obtain is that here it's the number of images that are used for the training. So maximum number, 89, and one image only for training. And what we can see is that with only 10 to 15 images, we obtain the maximum performance with genetic programming. It's not reaching CellPose level, but it's very close to CellPose level because, with very few images, because it's assembling functions that are engineered to do segmentation from that. So you don't need to reinvent Sobel filter, you don't need to reinvent Laplacian and everything inside the neural network. You just have to know how to use and optimize this function here. And we cannot see on this graph, but we obtain very good generalization capacity. It means that this is the test, and the train is very close to the same performance. On this same paper, we tried to segment tumor. So this is a melanoma tumor. So this is the image that you have in input. This is an annotation that has been done by a pathologist at the University Cancer Center in Toulouse. We have 24 of these images here. And what we can apply the exact same algorithm with the exact same function library to these images. So we trained on 12 images and kept 12 for testing. And you can see test images here. And because it's also quite fast to apply, and you have a lot of randomness in genetic algorithm, you can train many different models. And it's what we did here. We have 100 models that are trained and giving like a heat map of certainty, let's say, of the model, of the 100 models that are voting here. So you can see that it's working pretty well. I have the numerical result in the paper, and it's doing better than that started because specifically trained on this data set. What is interesting is that if I take the best pipeline that has developed, which is displayed here with so we pass this image here with the different channel. We can see all the step, and we work. So this is the immunologist that is working at the cancer center and the PhD student. And if we work together on this pipeline, we can have an explanation on how it was segmenting the images. So I will not read the full text, but to give you a sense, the biologist told us that it's using this channel. I don't remember which one. I think it's this one here to tell that the pipeline is looking at immune cells to get a first idea, and it's processing that until having a good vision on where the immune cells are. And then it's doing the difference with this channel here that is providing a big idea of where is the tissue. And by assembling these two, it's able to make this good mask of the tumor. The idea here is that it's necessary to have both the biologist and the image processing expert together in order to understand that, because I need the biologist to understand what's the biological reason to look at one specific channel or applying one specific operation, but I also need the computer scientist to explain what a Laplacian is doing to the biologist and to understand what's exactly the process. But because we have this readable pipeline of abstract function, but that I know what are doing, I can follow the decision making here. So then we went to decomposition by type, and basically it's the idea to follow how the different types of data are processed. And in medical data, it's very important because you have very different type of data, clinical data that are scalar or categorical most of the time, biology, so the blood test basically that you get, images like we saw just before, now more and more genomics, and each type of data requires specific processing function. You don't process genomics as you process images, for example. It's a different way of processing the data. And usually what a neural network is doing is that you have your different modalities, you put that into what we call an encoder. You take the scalar that are at the end of the encoder, you put them all together, and you have your classification head, your decoder, whatever, at the end. And actually, you very quickly lose where your data is coming from and how it's processed. So you have a loss of semantics of your data at the end. What we did, so the one big limitation that we had in the previous approach was it was limited at only one type of data, images. And what we did is to extend that. And the idea is that in evolutionary algorithm, we can have what we call different chromosomes. And the chromosome here will encode for a graph for each different type of data. So for example, here, it's a graph that is able to process vector of integers. Here you have a graph that is able, and a function library that is able to process strings, and another one for vector of strings. You could have another one for images, for genomics, for all different kind of data. And what we propose is the multimodal adaptive graph evolution, where all these chromosomes can be crossed with the type-safe mutation, meaning like when I mutate this function here, I know which kind of function library I can handle, because I know this function 12 here is only processing, for example, here, vector of integers as an input. So I know by definition what are the data types that are allowed in the function, and I can navigate in my genome in order to do all the right connection safely from the type to another one. I'm back to this slide here. What we obtain is that we have graphs that can take directly the inputs image to scalar values. So here I have a function that is transforming this image into a scalar, counting the number of colors that are inside the images, and I have 79. That gives me a number. And here I have image to image, so I can see this one is transformed by a morpho-Laplace into an image, and I can follow all the image part before being transformed into a scalar. So I can follow how the data types are processed before being used, and how it's used for the full decision process. And that's something important. I'm putting back again the doctors and the IT, the computer vision expert that has been used. We can explain how it works. Again, that's this story about the density of cells, because we have the doctors that is able to follow the data type process. So until here, how the image is processed and how it's transferred to a number. That is helping a lot the doctor to understand how the graph is taking the decision. So the big question for now that we are trying to target is how we can automatically decompose this work. So decomposing at the function level, I think we are in a good step. But how we can decompose that automatically? And actually, it's what we are doing in computer science. For example, this class diagram, here, you know, it's used to see all the functions and how they are linked together, assembled into classes, how the classes are linked. It makes you an idea of how your software is working. And it's the same in medicine. You can decompose the decision process in order to take the diagnosis for your tumor. You first segment the tumor, you look at the image and see where the tumor is, and then you are going to zoom in and see some characteristics, the number of division, the number of strange nucleus that you have in cells, and many other criterions that are used to take the decision. And then you aggregate all these features, let's say, and take the decision based on these features. And it's, again, what we are doing also in computer science here by trying to make our code very separate, very clean, and assemble that as Lego bricks at the end. The big problem that we are trying to target now is how to make that automatically done in genetic programming. The main limitation that we have is that for now we obtain very simplistic graph, like the one that I showed in this presentation. And it only allows for simple decision. Good enough for some data set, but if we want to target more complex decision, we need to have more complex graph. And actually, there are things that exist in the literature, especially back in the 2000s when engineering, I don't know what to say, génie logiciel, engineering, software engineering, yeah, perfect. Were a good thing and not anymore, not yet existing codecs and cloud and all the friends. We have metrics that exist to evaluate the quality of code, and it's used in the industry, in safety industry, when you in airplanes, in critical system. You need to have a certain thing that allows. And for example, we know how to decompose, to find the components that are in the code, how they are connected. So for example, this attribute here is used in this component here and here, and also propose automatic reorganization of the code that allows big blocks to appear that are bigger modules that can be reused and that are easier to understand because all the interaction will be in these blocks here. And I will not spend time on that, but there are complexity indexes that allows to do that and process that allows to reorganize the code or the engineering process in some design problem. So there are things that exist that would allow us to measure interpretability, and that is important for us in evolutionary computation to be able to measure. If we have a measure, it means that we can optimize for that because it's an evaluation function that we are going to pass to the algorithm, and I can do multi-objective optimization, efficiency to the task, so my classification task, and efficiency in my decomposition metric, for example. So in conclusion and openings, I think the explainability is not enough, especially in critical domains, because you see where the decision is taken, but not how it's taken. And this is the role of interpretability to understand the full decision process from the input, each decision part until the final decision that is taken. Interpretability is necessary, in my opinion, to make AI more acceptable. I think it's important for the people, the experts that are going to use this system to understand how the decision is taken by the method, by the model, because it's how they will believe that the system is taking decision for right reasons. The problem is that interpretability is a continuum. It's not binary. It's not interpretable, not interpretable. I showed that neural nets have the properties of interpretability, decomposition, simulability, but it's very hard for neural networks, and there are other approaches that are simpler. So it's hard to define if your method is interpretable or not. It's necessary to go to this exercise to interpret the models that you obtain at the end. Another problem is that it's human dependent. The interpretation that I provide to a doctor is not the same than the interpretation that I will provide to a computer vision expert or to a patient, each one with the different background. They will understand the decision process differently, things differently, function that computer vision expert can understand, like a Laplacian. It's okay. For a doctor, it's acceptable. For a patient, I don't think so. So it's very different, it's very specific to each person. That's for the optimization part. How we can optimize for interpretability. So this necessity to have metrics for interpretability in the domain. Currently, it's the size of the graph. And I quite disagree with this kind of metric. Being small doesn't mean that you are interpretable, and it pushes to small decision and simpler decision. Another idea, and that might be for you, interpretable is also dependent of humans. So if I optimize interpretability for me, it's not the same than for the doctor. So maybe we should put the doctor or the final user in the loop during the evolution in order to optimize for this specific interpretability. And what I believe is that if we have, and that's very opening, let's say, if we have interpretable models of whatever kind of data, of my classification, of my patient cancer, no cancer, then I can find new ways to take decision. And it means that I can ask question, why the model took this decision, how it took it. Maybe it's something that we didn't find before. That could be a new way of thinking about the problem and leading to potentially new scientific discoveries. It's something that in the community we are pushing, because if we have a model of the data that we can read, it means that I can understand how the model is this decision. And this is the team from a few years ago. It changed a little bit in terms of people, but all this work was only possible because of the big team that is there in Toulouse working on all this subject, and I'd like to thank. And thank you very much for your attention.
Thank you very much, Sylvain, for this very interesting presentation. Is there some question in the attendees? Yes, Nathan, go ahead.
So the genetic algorithm models, they combine filters, if I understood correctly, well-defined filters that have been made by humans. And also I've seen when you compared with the neural network performances that you slightly underperform it. And my question is, do you think genetic algorithm models like such are condemned to underperform neural networks precisely because they use tools that are well-defined and they cannot kind of explore and reinvent the wheel in their own way?
That's a very interesting question, actually. In most of the dataset on which we applied this method, we were slightly underperforming against neural network. Very recently, it's submitted. It's only submitted. It's not accepted anywhere yet. But what we show is that if we increase the number of program, like if you have a forest, let's say, of programs, like in random forest, a forest of decision trees, then you can converge to the number to the performance of neural networks. And we show that it's actually a balance of how many programs you are willing to interpret to obtain this performance. So if you are willing to interpret, like, let's say, 5 programs, then you are at 85% of the efficiency of your neural network. If you are willing to interpret, I don't remember exactly the numbers, but 20-ish programs, then you are 90%. And if you are willing to do 1,000 program, then you are at the performance of the neural network. So we can reach neural network performances, but then it means that we have a lot of programs to analyze. So it's really a balance to find between interpretability and performance.
Couldn't we like combine the two approaches? Like, for example, we first like use genetic programming to find the big blocks. Then we replace each block with like a neural network that approximates the block. And then we fine-tune the neural network using gradient descent to like optimize the cooperation between each block or something like this.
But then you lose interpretability because your function, like the Sobel, well, so the Sobel, it's not a good example because it's a convolution. But let's say a Laplacian, if you are using a neural networks to approximate, you don't have a Laplacian anymore. It's something that is looking like a Laplacian, but not exactly a Laplacian.
But I think we still have some kind of interpretability. It's simply that we use something that is similar to it,
we didn't try it, but what we thought at some point is to do the opposite. Like we have neural networks. If I go back to CellPose, we have neural networks. We know what they are doing. For example, CellPose is doing cell segmentation, and that could be a block that we assemble with something else. Like I know that I have this neural network segmenting cells. That's a function, and I can assemble this neural network with another thing. So it's the other way. But yeah, I think there are work in the literature, in the GP literature also, to build automatically machine learning pipelines, the auto ML way, let's say, but use GP in order to build machine learning pipelines and many different things. It's a big community currently in evolutionary computation in general called EvoML, where you're trying to combine the benefits of machine learning with the benefits of evolution, genetic programming, or others. I think there are many hybridation between two worlds that are possible and very interesting to explore.
Other question? One online, yes?
Yes. Question from Carlo Castoldi. So he says, amazing work and presentation. Thank you. Did you work into making your models and their training available on common open source software used by biologists, for example, QPath or Fiji?
So not directly, the models not themselves, to my knowledge, at least. The algorithm to generate these models are available. For example, this one here, this work here, the Cartesio software, we call it Cartesio, it's available. So you can download it and train it on your own data set. I don't think we have released the model, but that's something that we could do because that's the way we are working in the team. Thank you for the question.
Other question? Maybe I have one. If I correctly understood your presentation, in fact, you are using genetic programming, but when you are using genetic programming, you are not using traditional simpler operator, very basic operator like plus, minus, and so on. You are in fact using very high-level operators as a function of the domain on which you are applying genetic programming.
Yes.
Okay. My question is, so I correctly understood, it's nice. My question is, when you presented the graphs on the function of the results you obtained, what you were trying to interpret, yeah, for instance, this is it the resulting trees that you obtain from the GP application for this graph? This is a result of the.
Yes, that's one of the graph that has been generating with GP using the library that I was showing. So if I use this function library, I apply it to this data set here of images, I obtain this graph.
Okay, so
this is not the best one. For this paper, I don't remember. I think it's the third best because the first one was not interpretable, like it was multiplying two images, like multiplying pixel by pixel, so it doesn't have any meaning. Well, I mean, we didn't find any meaning. This one is interpretable, the third one. So, but yes, it's one of the graph that has been generating by GP.
Okay, so it is quite simpler graph compared to traditional problem in GP called the bloat. The bloat. No. So there is no bloat in.
So that's a property of the approach that we are using, Cartesian genetic programming. You have a fixed size encoding into the genome here. So you define how many genes that are allowed. And for example, usually we put 20 per function library, and it allows you graph that of small size in comparison to GP trees, where you can bloat because you have an infinite number of branches that can be created and tend to increase in size because of crossover and mutation at the end. Here, you know that your graph will be limited in size. And that's a very nice property when you try to interpret with this Cartesian genetic programming approach.
You mentioned just in your answer that it wasn't the tree that you were showing is not the best one. So when you obtain the results of the trees, you have to make manually the you have to evaluate manually the interpretability of the results of the genetic programming. So that's why you need or you want to go towards a measure that is able to measure the interpretability of the results. Okay, so it could be next multi-objective approach.
Yes, definitely. So for now, for example, this graph here, it was 2 days, I think, of work for my PhD student with the doctor in order to understand what he was doing exactly. To
analyze the
third one that was explainable. So it's a lot of work in order to understand how they are working, and that's a problem for now. And yes, if we have a measure of interpretability, but again, this measure is hard to find because it's dependent. The interpretability that you have as a computer scientist will not be the same as a patient, different from a doctor. So we all have different interpretation level depending on our background. But yes, if we can measure that, I can specifically optimize the graph to be both performant for the classification task and interpretable for the doctor, if I have a metric for the doctor, for the patient, if I have a metric for the patient, and for the computer scientist, if I have a metric for the computer scientist.
I have difficulties finally to understand why it is so difficult to know if the resulting trees are so difficult to interpret. You say it is 2 days or 3 days of work for interpretation, but why it is so difficult? Because the trees are quite simple.
Yeah, this one is very simple, but we have bigger ones that requires more time. So this one, I don't think this one specifically was 2 days, but on other problems that are bigger, we can have 2 days of work to understand how it's intertwined. I think it was on the Atari tool where I think I have a slide about that. No, I don't think so. No. But
maybe.
Yeah, but the problem is really to have in this specific case, to have the knowledge both from the medical field and from the computer vision field, because you cannot understand why it's using hematoxylin if you are not a doctor. So now in the team we know, because hematoxylin is a marker for a nucleus, so that you can acquire that with time. But it's the doctor at the end that is helping a lot. So it's a very interdisciplinary work to do at this point also.
Maybe a larger, not a larger, but a more question on reflection. You mentioned you are presenting very good works on images, but what about other domain where there is no images?
Yeah. So
is it possible? Is the approach is also applicable in other domains where there is no images? Because when you show us the difference between the explainability and the interpretability, you say that in fact the explainability is just a heat map of what has been used for the model to predict, but it is very easy in the case of image. But what is it so obvious in other domains? What is your opinion on that?
So it's a tricky question, this one. If you are thinking about tabular data, you have the same tool that are existing, feature importance, Shapley values that are showing where the decision is taken, the features that are used. Shapley value a little bit more, you know what's the influence, let's say, of the value of your feature to the decision inside the decision process. And you can use GP to compute numbers, and you have an equation at the end, and you could do the same work of interpretability. The big issue is usually you have a big equation that are harder because we don't, most of the time, most of the domains, we don't have domain-specific operators to combine these numbers. So usually you obtain big equations that are harder to interpret.
Okay.
But you can have the same thing with tabular data. I guess with genomics, if you have operators that are able to process your DNA or whatever, extract some, for example, some genes and how they are combined in some way, then you could have this idea of interpretability as well. So I think for each type of data, you should, an applicative domain, you should be able to find a domain-specific function library, because it's had been done in the past in research, in engineering. And the question is how you combine that in order to obtain the best decision process at the end.
Okay. Thank you, Sylvain. Is there, yes, one more question?
It's a reaction from Carlo Castoldi from your conversation a few minutes ago. So maybe, I don't know, I can either read it or you can.
Just maybe read it because it's quite long. And
okay. It would be very interested in that. In our lab, we have developed a pipeline for whole brain analysis. We use we use it to analyze batches of hundreds of high-res images for a total of about 1 or 2 terabytes, often requirements and tissue stain change, and so segmentation. Models must change as well, having models that learn with fewer labels, than common ML models, CellPose or instance egg, is very intriguing. Yes, that's also something that is interesting, is that you can have whatever kind of inputs. Here we have, we started with RGB, so the different channel, the grayscale R, G, and B. For a lot of time, we use also U segmentation, HSV space. And the doctor told us that there is a deconvolution of RGB to HED, which is the markers that are used for this specific type of image, that we added to evolution, and he used that at the end because hematoxylin is used. And it came actually also from this idea with this biologist here, Salvatore Valituti, is a biologist, is spending millions of euros to acquire very few images, like 100 images, and then deep learning cannot do anything if you don't have too few images. And that's where this approach started. It's because you have very specific images, very few of them, and you want to analyze that automatically, objectively, as much as possible. So in this case, we are starting to work also in the brain with in comatose and stuff like that. And I think it can apply very well if you have function libraries that are well done for this kind of image. I think it's a good applicative field.
Thank you, Sylvain. There is one, maybe the last question. Two questions. Oh, a lot of questions there.
Yeah.
So,
okay. So I've seen a presentation in physics, like in cosmology, in which they have very few images because they only have one image, and they use like models to synthesize images. Like they use like the image of the sky. They learn the statistical properties, and they generate images with same statistical properties. Like, couldn't we use the same kind of like approach to like generate a lot of images and then train our models on this?
Yes, we can. So I can't say much more for now about it, but we are starting to see. So it's called synthetic data generation. I think, and we have early results that I didn't present here, that shows that we can generate synthetic data that are close to the quality of neural network, to get back to one of the questions. So we don't know yet because it's very early, but with hopefully a model that can show how the data are synthesized. So meaning that if you have an interpretable model that is able to generate synthetic data, you can understand first how the data is working, and also be sure that the synthetic data are generated for good reason and with good basics, let's say. So it's a potential area of application.
The other question is like, how do we benchmark like CellPose, for example?
We benchmark against CellPose. For me, deep learning in many data sets, or machine learning, not always deep learning, but on images, it's deep learning currently. Deep learning is the best approach. If you are looking for performances, if you have big enough data set and you don't care about interpretability, use deep learning, don't use GP. You're going to be better than in terms of performance. But what we hope is to reach the performance of deep learning, beat deep learning one day. Why not? We can dream about it. But keep this, I mean, my main line is interpretability here, because I prefer to lose 1% of performance if I have an interpretable model that my expert, my user, is going to believe in and use at the end, because the expert knows that how the decision is taken is for a good reasons.
I mean, like, how do we evaluate that?
So here it's a segmentation task. I'm taking the best model of the literature, CellPose at the time. Now it's CellPose 2. They have metric, average precision, so it's the number of pixels that are inside the mask and the number of pixels that are outside the mask, basically. I have the exact metric, the exact same data set, the exact same split. So very comparable. I was going to say a scientific comparison between both of them, and I evaluate. And because it's fair comparison, thank you. And because it's evolutionary computation and we are subject to randomness, because the mutation and crossover process is very random, we do 20 runs, here 40 runs, I think even 40 runs. And it's why you have this standard deviation here to make sure that we are always converging to good solution, and the green dots here are the best model that we obtain. But we do robust statistics around this work, or around these runs, sorry.
But then you don't know that you are worse than CellPose. You just know that you are near CellPose. If you suppose that CellPose is the correct solution.
That's the current best for segmentation, for nucleus cell segmentation. That's the current best in the literature. Yeah, and I can, if you are doing denoising, the current best is diffusion model. That's the current best. So I'm going to compare to that. If you are doing synthetic data generation, it's diffusion model again, and you can do OR autoencoder. Then I'm going to take the best model, train them on the same data set, or use their data set and evaluate my method against them. And I don't know, maybe I will be bad, but it means that I have room for improvement. But I will publish when I will be close to.
Thank you for this very interesting discussion. Maybe we can continue with the coffee break. So this will end our session for today. The next session of the forum Numerica will be on the 7th of July with Giulia D'Angelo, who will present to us the neuromorphic computing and robotics. Thank you, everybody, for being there and being online also, and see you next time.